Jeremy Miller. I'm a scientist at the Allen Institute for Brain Science. And today I'm going to talk about different methods for network visualization and gene expression analysis. Oh, it's only for the camera. Should I speak louder? Okay. Okay. And so a lot of what I'm going to talk about at the very beginning of this is going to be stuff that you've already heard Monday and yesterday, but hopefully the more you hear it, the more it'll make sense. But stuff towards the end will be things you haven't heard yet, so if you're going to fall asleep, do it at the beginning of this part and not at the end of this part. So the outline for this talk is, first I'm going to talk about some visualization, how you can visualize networks using R. Then I'll discuss methods for visualizing networks using outside programs. And I'm going to focus on Vissant, which I'll talk about when I get to that. After that, I'll talk a little bit about gene enrichment analyses using R, and then some gene enrichment analyses you can do, or pathway analyses using outside programs. So to get started, if you want to visualize networks using R, you will first want to run your network, do your weighted gene co-expression network analysis, and then assign your modules. And as you've already heard, this process will involve creating a dendrogram, and the dendrogram is something that will show the topology of the network, although it doesn't directly show gene expression relationships or module correlations. And so what you want to ask once you have these modules in this network is, what do the modules represent? Which of these modules are distinct? And and if there are some similar patterns, if some have similar patterns. So, so here.
Appear jumpy to everybody?
Yes.
Okay. I don't know what we can do about that.
Well, ignore the jumping of the screen, and I'll continue. So, So a very useful thing when visualizing modules is to calculate the module eigengene, which, as we discussed, is the first principal component of the data and is a good representation of kind of a representative value of what the module represents in terms of gene expression. And a lot of the visualizations and enrichment strategies that you use will require using this module eigengene value. And so one thing you can do with these module eigengenes is you can visualize them as dendrograms just like you can with the genes. So down here would be an example of a network where you have your 8 different modules displayed as a dendrogram with the distance on this axis. And here is the same sort of idea, except it's showing the module eigengenes. And so modules that are in the same branch of this dendrogram would represent ones that have relatively similar expression patterns between these genes.
Sorry, Jeffrey, can I interrupt for a second? If that's bothering you too much, these slides are also posted on the website, so under the Wednesday lectures. So if it's easier to look at it on your computer screen than up there until they get it fixed for us, you can do that.
So the comment is these are also available on the website, these slides. So I'll wait a minute until they come back on the screen. Does anyone have any questions about the first slide? There we go. All right, much better. So one thing to comment is So these module eigengenes that are in the same sort branches would represent modules with genes that have relatively similar expression patterns. But I should point out that the genes within a given module will still have more similar expression patterns than the genes between these relatively similar modules. So another way of visualizing these modules is to use the multidimensional scaling plot. And this is a good way of plotting the first two principal components of a distance matrix, or you can also do 3 dimensions, which sometimes works out better. And as with the previous plot, the modules that group together on this plot will tend to contain genes that have similar expression patterns. So in this example, we did a WGCNA study looking at Alzheimer's disease, and we found these 4 groups of modules that represented different processes. So this one was 5 distinct groups of genes that all had some relationship with synapses and synaptic formation, another one with cell metabolism, and so on. And so this is often a useful way of displaying the data. So another useful visualization is looking at the module eigengenes directly, for example, using a bar graph. And in this case, each bar would represent a sample. Each bar would represent a sample, and it's important to order these samples in a way that makes sense. So this is the same data on the left and on the right. Here it's just the module eigengene without any labels plotted sort of randomly, and you get no information out of it whatsoever. But if you sort them and put some labels on there, you can see that this module represents genes that are higher in some regions of the hippocampus, CA3 and CA4, and may have a little bit of a gradient in this one, although maybe not as much. And bar plot, as well as the verbose bar plot in the WGCNA library, are 2 good R functions for plotting bar plots. And there's similar functions for scatter plots and box plots as well. And there are several other visualizations that you can use in R. These are, I guess, what I would consider more of the standard ones. So you can make heat maps where, as was mentioned yesterday, this axis would represent different genes color-coded by the module that the gene is in, and then the columns would represent different samples. And I don't know if these are sorted, but if you sort the samples, you can often see a block-like structure in here, which would show you the different modules. Yes?
Could you remind us what the R commands are, and what is the p-value above the box plot?
So the question is to remind us what the R commands are for these and what the p-values represent above the plots. I believe there's one called heat map to make the heat map and plot mat. Okay. The scatter plot one is the verbose scatter plot function in the WGCNA library, and above the plot, the part of the output is the correlation of the Pearson correlation of the genes that are in or whatever you plot in these 2 axes, and the p-value is the p-value corresponding to this correlation, so the significance of the correlation that you get back. The one on the right, this box plot, is the verbose box plot command, and for this, as well as for the bar plot, you get back a p-value that corresponds to, I believe, a Kruskal-Wallis test that tells you the significance of differential expression between either 2 or if you have more than 2 boxes that you get back from the plot.
What's the meaning of the notches and the horizontal line?
So I believe it's the median and various quantiles are displayed. The 75th and the 95th quantiles are displayed, and then the point at the top would be anything that's above and below the 5th and 95th quantiles of the plots.
The notches are the 95% confidence intervals of the median.
So the notches are the 95% confidence intervals of the median. Any other comments on this or questions? Okay. So this is more of a general comment slide that it's very important to get your data visualizations in a format that are appropriate for whatever the data is that you're looking at. So I found that for the really complex data sets that I've been looking at, that it's often useful to have compact summarizations of the data, and as I mentioned before, to properly order the samples in a way that makes sense. So here, this shows 150 bars corresponding to 150 different brain regions. There were about 900 samples that were run in this particular brain, so having 900 in a row would be a lot more. I mean, even already 150 in a row is pretty long, but 900 in a row would be much harder to see. Likewise here, I want to show a gradient in the brain, so I sort of overlaid a picture of the brain onto these data to give you an idea of where these samples come from. And here, this representation here is about 100 different, or I think it was more like 500 different samples collapsed into a small box that I think gives more of a general idea of, you know, the laminar expression and the changes with age that you would get if you plotted it all sort of linearly. So just visualizations are important, as has been mentioned several times already in this course. So there are several other ways of visualizing outside of R. There are a few programs that do both enrichments and visualizations, and I'll talk about those when I'm talking about enrichment. So Chilibot, Ingenuity, and String are three of these examples. Vissant is a program that will let you plot out your modules, and I'll show you that in the next slide. It's available for both PC and Mac. I haven't tried it for Mac for a couple of years, but the last time I tried it on a Mac, it actually worked better on a PC, so just keep that in mind. And then there's Cytoscape, which has been extensively discussed already, and it's another, it seems like a very, very good option for plotting modules. And I did want to just point out the command export network to Cytoscape, which will allow you to put things into Cytoscape if you wanted to, and it's available in the GSEA library. So if you want to use Vissant, what you would want to do is download it from the website and download Java as well. Then you will need to create a file that has all of your interactions in the appropriate format, and I will shortly discuss what the appropriate format is. You'll then want to read your interaction data into Vissant and then format your interactions map in Vissant to make them look however you want. And this would be an example of a module that's plotted in Vissant, where these larger hub points are the hub genes, and these smaller ones are the other genes in a given module. And I've put together a little tutorial about how to use Vissant with different screenshots, and it will tell you how to do exactly this and nothing more, nothing less in Vissant. So if this is what your goal is, then Vissant is a good thing to use. If you want to do other things beyond that, then there are other programs that will be useful. So there are three different ways of making the appropriate input file for Vissant, and you have to have it in the appropriate format or else it won't work. So one of them is you can just make a file on your own, and it will have to be in this format over here. The export network to Vissant command in the GSEA library will take your data and put it in this format automatically. There's also another function that does essentially the same thing that's in the meta-analysis R object that I was discussing yesterday. And so in the file, you basically have the two genes involved in the interaction. I'm not sure if these are directed or not. I've only used undirected interactions. You then need a column that I just have the number 0 in. You need a column corresponding to the color, and you can't just put color names in. There's a code system which corresponds to different colors that I think you can find on the Vissant website. And then you have to have a number which can be used for various filtering things. Usually I use the topological overlap here. So if you, for example, only wanted to show the connections that had the strongest gene-gene interactions in a module, then you could use this number. And so then you would just copy and paste whatever you wanted from this table into Vissant. And this is where you would paste it into, this is what a screenshot of what Vissant looks like. So you'd paste your data in here. Over here, there's an option called Fine Arts, which adjusts the thickness of the lines. I don't like having them turned on, but that's just a personal preference. You can also change things like whether you want to have the names of the nodes, the size of them, various things like that under the properties, the nodes properties panel. The layout here is very similar to what was being discussed yesterday in Cytoscape, to make it automatically move the things around to make it look like this rather than just a big mess. Once you do that, you'll probably have to manually adjust things. This is where you would look for, if you wanted to adjust your weights so that only certain of the connections are shown. And you'd probably want to use this last, because if you remove connections and then do your layout, it'll send things way off the screen, which can be problematic. And then finally, you'd want to save everything as both the image and the Vissant file. And that's how you would make something like this. So moving on to gene enrichment analysis, there are two basic methods for gene enrichment analysis in R. One is that you can do enrichment for published or user-defined lists, and this would be using the user list enrichment function in the WGCNA library. And this will perform hypergeometric tests for all of your modules or any lists you want against any user-defined lists. And this also will include some preloaded lists from brain, blood, stem cell data sets, as well as a few other things that they're continuously being updated. So some examples, oh, go ahead.
Yeah.
Okay, so some examples would be cell type markers from many different publications. You can have genes from modules that were published in earlier WGCNA analyses. So for example, modules associated with autism or various areas of the brain. Also known and predicted lists for disease genes, list of genes enriched in particular brain areas, or immune-related gene lists. And if you have any lists that you think would be important for the community, you can certainly let us know and we can add them to the function. Also, you can do the second, yes?
I just wanted to echo what Jeremy said about user list enrichment. This is a tremendously valuable function for annotating modules. Jeremy doesn't highlight it enough because he's the author of the function and he's very humble, but let me mention it can do things that other tools cannot do, especially if you have neuroscientific applications, because it contains gene lists that were assembled on the one hand by Jeremy, things, for example, a list of Alzheimer's disease-related genes or autism-related genes. But also it contains hundreds of lists that are relevant for Huntington's disease, because the CHDI Foundation and Mike Palazzolo, they assembled gene lists for the last 15 years, and they are all loaded in here. And also it contains a lot of gene lists related to stem cell functions that could also be very exciting. And so, and plus we now have this effort to continually updating these gene lists, also with respect to important modules published in other articles. For example, Mike Oldham at some point characterized the module structure in the brain. And so all of these module assignments then are in this function. Same with modules from the Allen Brain Institute analysis of the human brain. And so it allows you to compare your modules to those that were found in other studies. So I encourage you to use it, yes.
Is there an overview somewhere of these lists?
Yeah, you just do the help file.
The help file, yes.
And there's certain options. So let's say your data come from blood, then there's certain gene lists that relate to blood. And again, very valuable. Example, there are sometimes modules that relate to red blood cells. If you remember, red blood cells don't have DNA, So, but we do find modules enriched with red blood cell markers, and so you would be able to detect it. There are lists related to CD8 T cells, CD4 T cells, B cells, and so on. And so this is a very useful function that can do things that other functions, other tools are not tailor-made for WGCNA.
So the general comments there was that, for the camera, is that it can do, it can also look for things like Huntington's disease, different stem cell markers, and a lot of other things that you can't find in any other functions. It's very valuable, and if you want to know more about it, you can look at the help file in the WGCNA library, or in R. For user list enrichment. So the other type of enrichment analysis you can do are gene ontology enrichment. And there's a Go enrichment analysis function in the WGCNA library, and enrich Go in the cluster profile library, and there's about 20 other ones that you can find on Bioconductor. But in my experience, I found that the results that you get from functions outside of R, like David or Ease directly, tend to be better than any of these R ones that I've looked at least. And I should also point out that there is in the works an R function combining all of the above that will hopefully be available soon. Mm-hmm.
You mentioned that the David is better. Is it because the result more accurate, or they're just better in visualization?
So the question is, why is David and Ease better than the ones in R? I'm not exactly sure. It seems like the categories that you get back are ones that are you get more of them back, and they tend to be more relevant than the ones you get back from the ones in R. It's also faster. I'm not sure why it should make a difference.
Bigger support, you know. Think about David East,
Yeah.
they have a huge team, you know, whereas R may have the occasional programmer.
Right.
Having said this, our Go enrichment analysis function is regularly updated, so you won't miss much by using it.
So there's more support for David and Ease, but the Go enrichment analysis function is also updated very regularly.
For the user list one, you mentioned that we can submit lists to you guys to add in. Can you just upload your own list to the function as well?
So the question was that you can submit lists to us, but can you upload them on your own? And the answer is no. You have to email them to us, and the next time the WGCNA library is uploaded or is updated, we'll add your lists. So, for example, we don't have anything related to plant biology right now.
But we will credit you. If you agree, there will be a citation added to the help file. So whenever people report that their module is enriched with your favorite gene list, they will then know who to cite for it.
Yes. So that's the comment was that all of the references that are that use lists in user list enrichments have the references in the help file. So anyone who looks at the help file will see where every single thing that they get in the output comes from directly. So there are there are many outside programs for gene enrichment or pathway analysis. I'm just going to very briefly discuss the slide on some of my favorite ones, but this by no means represents all of the ones that there are available. And you can come back to this slide later to get the actual links if you're interested. So the first one is EASE, or Enrichment Analysis Systematic Explorer, which is a standalone version of David that can be used to find enrichments for gene ontology, KEGG pathways, SwissProt keywords, gene map pathways. I think it also does different chromosomes. And then it can test the enrichment in a given test list against a reference list. There are several different options that you can use. I find that it's useful to output to save and output the data to an Excel file, which would be up here. The default is to send it to a web browser, which has the advantage that you can click on the links and it'll tell you more about the gene ontology and the other pathways. But the disadvantage is that it's not saved on your computer anywhere. There's a box here called Select Categorical Systems, which is very important because that will tell you which of these systems are actually going to be included in the enrichment. So if you're only interested in biological processes, you would basically uncheck everything else. But if you want to just find everything, you'll have the advantage of finding more things, but you'd have the disadvantage that you would increase the number of tests you're doing, so you'd get fewer things back that were significant. But yeah, this is a very useful program to know. Really the only disadvantage that I can think of is if you have a lot of lists you want to compare, you have to do them one at a time. There's not a way of batch doing comparisons, so you'll do a lot of copying and pasting if you have 10 or 20 modules you're looking at.
Why is it better than just using David directly?
I don't think it is better than using David directly. I think they're both equally good. I just prefer to use this one because I can have it downloaded on my computer. I think David has more updated lists than EASE does. You have to download them separately onto your computer to use them with EASE. So the question was, is EASE better than David, or is David better than EASE?
Well, it seems like as soon as you download these, it becomes obsolete the next day if data
Yes.
is right, updated regularly.
Right. So the comment is once you download ease, it becomes obsolete right away because David is updated regularly. I think that may be true. I prefer to use ease, and if you keep on top of downloading the updates from David, then ease will work just as well. But that is a good point that for ease you'd want to download the updates that come on David. It doesn't happen automatically. Okay. There's another similar enrichment analysis tool called Topfund, which I like because it's a user-friendly website and it provides gene enrichment annotations based on all of these various things on the left here. There are also several tools on this website that will allow you to do candidate gene prioritization. I haven't really tested them out, but I've heard that they're useful. The advantage of this one is all you do is you copy a gene list in and hit submit, and then you get back a bunch of stuff. The disadvantage is you can't put a reference list in, as far as I can tell. So if you're doing a comparison with only a small percentage of the genome, then the reference is going to, the things you get back might not be as accurate as they would if you could put in a reference list, because the reference list is very important. Chilibot is a literature search tool, and so this will take a list of up to 50 genes, or it doesn't have to be genes, it can be any terms. You could put in 45 genes, then 5 things related to the biology of those genes if you wanted to. And then it'll search the literature for co-occurrences of these terms, and then it can output an interactive plot of literature connections between the terms. So this would be an example of, I think I put 20 genes in related to the overlap of 2 modules. And you can then click on these connections between the genes to get back the relationships that were found in the text. And the colors of these would represent whether it's, they've called it a positive or a negative or a parallel interaction. One word of caution here is, as with any literature search, you should check the references carefully because, as was pointed out yesterday, if you say, gene A and gene B are not interacting, then it'll still show up in this, but that will not be a true connection. But I found this is a useful way to kind of quickly get an idea of whether the genes in your module are related in the literature. Web Gestalt is a toolkit is another toolkit for enrichment analysis, and this can perform several enrichment analyses from a relatively straightforward web-based interface. I haven't used this as much as some of the other ones because I haven't found the output quite as user-friendly, but there have been a few cases where I haven't been able to find anything from the other tools, and Web Gestalt gave me some interesting results. And I'm not sure if it's because they use different types of lists that go into their enrichments, but sometimes this will come out to be something more interesting than the other ones. So there's a program called Ingenuity, which is a hand-curated list of interactions. This has, I think there's like 50 or 60 PhDs that work in this company, and they go and look at all of these papers and hand put together different types of interactions and what kind of interactions they are, whether they're protein-protein interactions, whether gene expression interactions, and the direction of the interactions. And you can get back these networks that show them all these interactions together. And if you put in a big list of genes, it'll give you back subnetworks of 20 or 30 genes each that'll show you things that are interacting together in the literature. This is very useful, but it requires some training and has a very expensive subscription to use. So if you happen to be in a lab that has a subscription to this, I would definitely suggest taking advantage of it. So gene set enrichment analysis, so GSEA, is a very powerful method for gene enrichment. The main difference between this and some of the other enrichment analyses is this takes into consideration all of the genes with respect to some parameter. So for example, correlation with age or module membership value. And it'll ask whether an a priori defined set of genes, so is significantly enriched at one end of the distribution. So for example, if you have, say, a list of, genes related to age, and you want to do a p-value cutoff, there might be some just below the p-value cutoff that don't get picked up if you use something like a normal gene ontology enrichment program. But with this one, because it includes all the genes, it'll take that into consideration as well. I believe there are a lot of lists that are put on the website that you can do your comparisons against as the defined sets of genes. I would imagine gene ontology is probably some of them. There's also ones from various publications that people have put on. This used to be pretty finicky about getting the data in the correct format for it to work properly. Like I had found cases where I would put it in the correct format and upload it and it would crash. But they've been updating it a lot recently, and I think a lot of those issues that I had a while back are very quickly going away. So now I think it's a very useful thing to use. Okay, so UGET is a tool for finding other co-expressed genes. I think that's hosted here at UCLA. It isn't really an enrichment method itself, but it can be very helpful for finding other genes correlated with your genes of interest across thousands of microarray samples in the Celsius database. So this is a very good way of doing guilt by association if you have a hub gene or two that you're interested in looking up. And so, for example, you could put in one or many genes in this first list, and then it'll find the correlation across all of the samples from a given microarray platform. Or if you wanted, you could specify a certain subset of samples you wanted to do this, or sorry, genes that you wanted to do this against. And it'll output this list of the genes that have the highest correlation with your genes of interest. So in this case, I put in a bunch of ribosomal proteins, and this NPM1 came out as the one of the top correlates. So this might be an example of another gene that's related to translation. One thing to note is a lot of the data on here comes from the Gene Expression Omnibus database. So a lot of the samples involved in the correlations are cancer-related samples, which may or may not affect what you get back. If you're studying cancer, that might be great. If you're studying other things, just that's something to consider when you get back the correlations. There's another similar program called String, which is a tool for finding networks of one gene. Basically, you'll put a single gene as input into this program, and it'll output a list and a plot of predicted functional partners based on several lines of evidence in the literature. So it's also not really an enrichment analysis, but it's very useful for following up things like hub genes. And I don't know if you can read what these things say down here, but it can tell you whether the genes in the interactions are part of the same neighborhood, if they're gene fusions, if there's co-occurrences, if there's co-expression, if there's from the same experiment, if they're from the same databases. It also does text mining and homology. So there's a lot of different things that come up as potential connections.
I was just wondering, in this tool, can you filter by whether you want experimentally validated interactions versus predicted interactions?
So the question is, can you filter in this tool whether you want experimentally validated versus predicted interactions? And the answer is you can definitely filter by some things. I don't remember off the top of my head exactly what you can filter by. So another tool that I'm just going to put up here as a first slide, because it does almost everything, is Galaxy. It can do enrichment analysis, and it can do like hundreds of other bioinformatic type things. It has a very steep learning curve, but there are a lot of tutorials on the main page that will help get you started. And from the people who I've talked to who use this a lot, they say it's really useful, but it takes a while to get used to it. So this is something you should definitely at least look into and maybe watch one or two of the tutorials to see if it would be something that would be useful. So are there any questions about any of those tools or anything so far? Mm-hmm. So the question is, are there any plans to integrate WGCNA into Galaxy? I'm going to defer to Steve. I have no idea.
Not yet, but if you want us to, do you recommend that we interface with them?
Yes.
Okay, good. All right, it's on our to-do list. No, in the back of my mind, I plan to do it. We'll work on it.
Just a reference.
Yeah.
So the answer, the question was there any plans to integrate WGCNA into Galaxy? And the answer is not yet, but it'll be useful, so it's kind of on the back burner now. So in summary, once you have your network, it's useful to visualize it. Once you have your modules, it's useful to visualize and annotate them to get a better understanding of what the genes represent. There are many different ways of visualizing and annotating modules, both within R and by using additional programs. And many of these methods will work with any gene lists, not just gene lists that come from modules. So if you have any gene lists that you care about, you can put them into these programs and get back your pathway analysis or your gene enrichment analysis. So that's all I have. Thank you.